start out with DNA molecule, and it unzips, but not entirely. It unzips in that fashion. Like when you have a broken zipper on your jacket, or your purse, your duffel bag, and you have a broken jacket on your backpack, it kind of splits in the center where it's not supposed to, and the rest of it is intact. But there's a gap, separated by a gap in the center. Very irritating. There's an enzyme that does this. It unzips the DNA. There's a genetic code along the DNA molecule. The genetic code has punctuation. Genetic code says initiate. So the enzyme attaches there and unzips. And then there's another genetic code that says terminate. So it stops unzipping. It only unzips from the beginning to the end of that gene. Only one gene is unzipped. And hanging, separated, would be genetic code. Thymine, cytosine, guanine, adenine, cytosine, guanine, thymine. I made that up. You understand? It doesn't mean anything. But on the other side, on this other loop here, there would be the complementary. The other half here, but I'm not going to write that down. One side is coded. This is the sense strand of DNA. Sense. The other side is just packing material. That's nonsense. Sense and nonsense. Only half the DNA is the gene. The other half is packing material, just to hold the place so that things don't fall apart, dismantle. It gives structural integrity to the DNA molecule, have a matching set of complementary nitrogen bases on the other side. So only one side of the DNA molecule is common. There's messenger RNA. I'm going to do this in blue. So this is going to be a messenger RNA nucleotide, one with cytosine. I'm leaving off the S and the P for the sugar and the phosphate. The sugar would be ribose sugar. And there wouldn't be any thymine. It would be uracil instead. So you got a bunch of these RNA nucleotides floating around. When the cell is involved in protein synthesis, RNA nucleotides are available from your food. Your food is made out of cells, cells have nuclei, there's nucleotides. So the matchings are just as exact as this. But there's no thymine in RNA, so there's a little bit of an alteration here. Attached to the thymine would be adenine of messenger RNA, adenine, thymine matched together, just as in DNA matching. Curved line letters go together, cytosine with guanine, guanine with cytosine. In messenger RNA, there is no thymine, but uracil takes its place. So where there would be a match between an adenine of DNA and something else, the messenger RNA substitutes uracil instead of thymine. So these are the specific pairings that occur in transcription. So this is replication. That's the pairings. And this is transcription. So, attached to the sense side of the DNA molecule, TCG, there would be messenger RNA AGC. And where there's adenine, cytosine, on the DNA sense side, it would be uracil, guanine. And where there's, well, anyway, I don't know. That's how the matching would go. And as the nitrogen bases line up, the sugar and the phosphate line up as well. And there's RNA polymerase that catalyzes the covalent bond formation between the sugar of one RNA nucleotide and phosphate of the neighbor RNA nucleotide. So this transcription between DNA and RNA takes place within the nucleus. That's where the chromatin is located. It's in the nucleus. So the DNA unzips a little bit and messenger RNA copies genetic code. So now you have messenger RNA peels off. So you have a single-stranded RNA molecule with adenine, guanine, cytosine, uracil, guanine, cytosine, adenine, adenine. That set of nitrogen bases from a single-stranded RNA molecule. That's the RNA transcription of the DNA genetic code. Question on this? This is messenger RNA. And you notice, the way I wrote it down, it's in sets of 3. That's because in the genetic code, a set of 3 nitrogen bases is the code for one amino acid. I'm making this up over here, but adenine, cytosine, guanine might be the code for lysine. I made that up, don't write that down. And the code guanine, guanine, guanine might be the genetic code for methionine. That's another amino acid. So where you see these sets of 3 nitrogen bases, you can translate to amino acids in a protein. I made this up now. Don't hold me to this. But there is a translation. You can do a web search, and scientists have translated all the amino acids to their triplet of nitrogen base in the code of the genetic code. They've solved it. It's deciphered. But it's not this. Don't memorize it. So messenger RNA comes off, and it is read. The code is read in sets of 3. That's 1 unit of code, and this is called a codon. Every set of 3 is called a codon. 1 unit of code. Because that's going to eventually result in a particular amino acid being assembled in the DNA, in the protein molecule. Okay, so that's transcription. Need black ink anymore? The second step, protein synthesis. Getting too low on the board. Second step, protein synthesis is translation. Translation is the assembly of amino acids into protein. The actual synthesis of protein is called translation. The code, the genetic code, is translated to a sequence of amino acids. And this occurs within the cytoplasm. This happens in the cytoplasm, not the nucleus. And it happens specifically at ribosomes. Ribosomes are a participant in the process of translation. So this is why the nucleus has nuclear pores, big holes. The messenger RNA is a pretty long molecule. It's 500 units in length on average. This molecule has to thread its way out of the nucleus through the nuclear pore. And then it goes into the cytoplasm. And that's the ribosome. Looks like a lopsided snowman. The messenger RNA threads its way into the ribosome. And you have the codon, A, G, C. That one is in the first place. And then there's U, U, G, and then there's CAA. So that's my messenger RNA. It's threaded into the ribosome like a piece of thread in a sewing machine. A belt of rounds in a machine. So the idea is there is a specificity between the genetic code in sets of 3 nitrogen bases to one of these amino acids. So wouldn't it be nice if the amino acid would read this and just stick on here, that's the first one, and the next amino acid with this code would plug in there, and then you'd have the 2 amino acids. And then the third one would plug into CAA, and then now you have 3. Wouldn't that be nice? Unfortunately, it's more complicated than that. There is another DNA, there is another form of RNA. It's called transfer RNA. Transfer RNA. T stands for transfer. And it is a single-stranded RNA molecule. It has on one end a complement to the codon. So this would be UCG. There is a complement to the codon. A matching takes place. The transfer RNA and the messenger RNA will pair the nitrogen bases with specificity. Attached to the adenine will be uracil, attached to cytosine will be guanine. Like so, the pairings will occur like that. The specificity is like pieces of jigsaw puzzle matching up. And when the match occurs between messenger RNA and transfer RNA, that is translation. So you know, on the messenger RNA, you have codons. There are 3 codons on this messenger RNA. What do you call a counterpart that matches the codon? You call this an anticodon. Anticodon is a set of 3 nitrogen bases on the transfer RNA that is complementary for the codon on the messenger RNA. So this guy, UCG, is an anticodon, will match up with AGC. The pairings are exact according to this table. And hanging off the end, the glycine. Could be anything, I made that up. So, could be anything, but UCG, anticodon, matches up, and glycine becomes the first amino acid in the protein making something. This messenger RNA feeds through, like a thread feeds through a sewing machine. This messenger and the next codon lines up, and it matches up with AAC on the transfer RNA molecule, and that's leucine.